Hey class. Today we talked about the immune system. In class and in lecture, we really tried to dive into the difference between innate immunity, adaptive immunity, and then with the 2 hours that we had in lab, we really hammered home about T cells, split into cytotoxic T cells and helper T cells. Now we're gonna close that loop on the last thing on adaptive immunity and look at B cells. So, B cells are all about antibodies. They have B cell receptors that bind to specific antigens. Like I've been talking throughout all class, it's that lock and key methodology in the adaptive system. Now, they are 3 different phases. First one, it's the activation. It recognizes the specific antigen, triggers it to change and secrete antibodies. After the antibodies are secreted, they are now in the blood system. Antibodies raise dramatically. You have immunoglobulins or gamma globulins, which are directly responsible for the actions that lead to the destruction of antigens when we bind, which we'll talk about it. And then the persistence of population B cells, memory B cells. This is talking about the memory response where you have the difference in innate and adaptive. Adaptive remembers. This is why when you get sick with something and you get sick with it again, your body reacts much faster to clear up that infection because it remembers. Now, this is going to be talking about in comparison to how that immature B cell, where it comes, how it matures, where it lives, and the process of becoming it. So, it develops in the bone marrow. Billions of B cells are produced a day, and only about 10% actually finish their maturation process. Remember, we're talking about any self-antigen or anything that the body can recognize as being found in the body must be destroyed. The reason for this is if it's not, we're going to get autoimmune disorders, and that's when things are going to start going wrong. So this is talking about the recombination thing, and let's walk through the process where the T cells needed to mature in the thymus. The B cells will actually mature in the process in the bone marrow before reaching its eventual home, the lymph node. So T for thymus, that's where T cells have to mature. B for B cells and bone marrow, that's where they are born and matured. Similar to the recombination testing, just making sure that you don't have a self-identified lock and key. Ends up in the lymph node. Now, remember, they are naive and mature. They are naive until they are activated. The B cell binds its antigen when activated. So let's talk about this activation process and what will eventually happen. So, boom, we're in the lymph node. We are a mature, naive B cell, and we get presented an antigen. Remember, if you're a B cell, chances are you're going to be presented with an MHC2 molecule. Remember, those are the binding complexes. There's the 1 and the 2. For the 1, relate it to you have a toxic X and can only see things in one way. MHC1, cytotoxic T cell, and they deal with intracellular infections. I'm talking viruses, I'm talking intracellular bacteria. MHC2, on the other hand, is going to activate and bind B cells and helper T cells, which is good to know because those 2 things are actually needed to make a B cell turn on fully. So we're in the lymph node. We have our B cell receptors, we have our antigens, we have our MHC2, our presenting, essentially, cell. We then take this sensitized B cell, and we take this T helper cell with this MHC2 binding site. Once those 2 pieces are there, that's when the magic happens. You can now see this sensitized B cell start dividing. First off, every time in adaptive immunity, when a B cell or a T cell is primed, it will go off into memory cells. And the memory cells are important because that's what will make your immune system respond much faster to the infection if they ever experience it or encounter it again. It's really cool. It's one of the reasons why when the common cold goes around and you get sick once and you're sick for like a week or a week two and you're like, and then if you get sick again, you're much faster to respond to it because your body already knows, oh, wait a minute, we have this B cell that we've already activated. We know this guy. Let's go show him this, and this worked last time. Will it work again? Spoiler, it will. So remember, in adaptive immunity, when that cell is activated, if it's a B cell, if it's T cell, some of it, as it's dividing, gets stored as memory cells. The other ones will then go off and do the function that it needs to. And in the case of a B cell, those that are going to produce antibodies are actually called plasma cells. So the B cells divided into plasma cells. Plasma cells are just going to dump antibodies out into the bloodstream because the antibodies are essentially going to be how the B cell is going to fight that infection. So now, antibodies are incredibly complex. There's actually a whole study and field of it, which is serotology. For the purpose of this class and this exam, let me tell you what you need to know. First off, understand the structural units of the antibody, where it is a Y-shaped molecule with 2 heavy and 2 light changes. Conceptually, understand that there's a constant region and a variable region. Now, it looks like this. The constant region is going to be the same. Think about this as your structural backbone. All of them are going to be very similar, but the variable regions, right at the tips, those green portions, those are your antigen binding sites. That is what is going to change depending on the specific lock and key mechanism. Now, earlier I have talked at length in class about this cell right here. If this is a plasma cell and its specific lock and key is AAA, that means that this antigen binding site is looking for AAA. Because, if you think about it, the antigen that was presented in the MHC2 complex to activate this B cell, at the time it was a B cell, now it's a plasma cell because it's producing antibodies, but originally when it activated, it needed to be presented with the AAA. Wow, it just keeps shooting to the left. Please ignore that. But the most important thing to remember is that specific antigen binding site is looking at its codes for whatever activated it. Why that's important? Let's find out. But that's what I want you to know about antibodies, bare minimum. You don't need to know into all the super depth about it, but you need to understand there's a constant region that's similar depending on what antibody structure you have. There is the antigen binding site, or the variable region. That changes specifically to bind a specific antigen. I have skipped ahead a few slides because, again, I like to work from the pictures and work our way back. Here you can see the 5 different types of antibodies with a specific function and a cool fact about them. Now, I won't be going super in-depth in terms of draw me the structure or what's the main difference between IgG or IgM in terms of the penta one and everything, but you should know that there's 5. And you should know some facts or at least some interesting concepts about them. So like IgG makes up the majority of the antibodies in the serum. You probably don't need to know that it is the only one that can cross the placenta from mother to fetus. It is an interesting fact to know, probably not going to test you on it, but just think about like the most common form of it and try to be like, oh, IgG is the most common. IgM is the first antibody secreted. If you think about it, it's because it's the biggest one, and since it doesn't exactly know what to target and how effective it's going to be, why don't you start with the biggest and then start producing after that big wave has already happened. Now, there are 5 antigens, and there are 5 antibodies, and there are 5 different unique effects of antibodies. The only one that is unique to a singular one is IgE. IgE binds mast cells and basophils and triggers the release of inflammatory mediators. We've talked about inflammatory mediators earlier in innate immunity, but think about this with specific and adaptive immunity. These antibodies are actually going to create a larger inflammatory response, which we've already talked about at length why that can be beneficial. Now, one of the big ones is agglutination and precipitation. Essentially, the antibodies are going to clump on the antigens and then try to stick them all together. This is twofold. One, it stops the bacterial infection or whatever kind of infection you're having to have the same level of motility or to have the same level of binding. If they're all stuck together, it makes it really easy for those phagocytes to eat it, because you're not just only getting one, you're now going to be eating multiple at a time. More efficient. Oops, scrolled. Okay. There's opsonization. So this essentially is, it binds it coats, and enhances phagocytosis. Again, the agglutination, precipitation, these essentially are both here to enhance phagocytosis or make it easier for the things to eat them. It also stops the binding abilities. Neutralization. So occasionally, the bacteria or the infection is going to secrete a certain type of toxin. It essentially is to either slow down an infection or to do something else to the body to make it more habitable or more survivability for that antigen. Now, neutralization. The toxin can't do anything if it's completely bound by antigens. This is or antibodies. This is blocking the effect, not allowing it to go and do what it wants to do. There is complement activation. As we talked about in class, there was the membrane attack complex, or the MAC, which essentially would create holes or perforations into your invader and then causing the cells to lyse and leach their materials out. Last one, stimulation of inflammation. It is going to help that inflammatory response. Let's talk a little bit more about memory. So memory B cells are responsible for antibody-mediated immunological memory. Sounds scarier than it actually is. Essentially, the second time that you are exposed to the antigen, you have phase 3 of an immune response. Now, the first time, you have a primary immune response. So when you are first introduced to the antigen, this is talking about the effective but slow method. It's why you are sicker for longer, because remember, when you're first finding it, that key has to go through every single B cell until it finds its matching one. And once that lock clicks, remember, then there also has to be the helper T cell lock click. Once those two things are activated, boom, then we get the memory cells that are going to stay behind, and we also get our plasma cells that are going to produce antibodies. It's effective but slow, and the antibodies peak about 7 to 14 days after it's encountered. That's when you feel sick. Now, future exposure is secondary immune response. So look at this. This could be a question. I could ask the difference between a primary immune response or a secondary immune response. The secondary immune response is after we have memory cells, meaning that we can skip the step that a helper T cell has to activate. It's a shorter lag phase. The antibodies peak more rapidly, and the peak is 100 to 1,000 times larger because the body knows at that point, wait a minute, this worked the first time. We didn't dump out as much and put all of our resources into solving that problem because we didn't know if it was actually going to work. Because it worked and because our host lived and we have this memory cell, let's dump it. So, how does this look on a graph or a chart? Primary one, there's a longer lag time, longer peak. The primary antibody is that IgM, or that pentamer, where it's that big circle one, because remember, the body wants to be as efficient as possible, so it's going to produce those really, really big ones to have an effect. Duration of response, 14 to 21. If you can see here, the antibodies levels rise slowly and at lower levels in the primary response. Secondary immune response, the lag time and the peak until antibody, much slower. The difference here is that IgG, it's that one that's most commonly produced. And you can see it rises rapidly in the secondary response. The IgM also works, but it's a lot less. And the reason for that is while the IgM is a more it's a higher cost antibody. Yeah, it has a lot more spikes and a lot more peaks and a lot more everything. It takes more energy to produce. Compared to with IgG, when you're dumping out tons and tons and tons of antibodies, you're going to get a huge response and it's actually going to be much faster. And if you can see duration of response, your body keeps it up because they want to make sure that you're protected. Now, humans are cool because we can actually use this method right in here, our brain, where we have actually tricked our immune system to work for us earlier using vaccinations. It's also called immunization, where we expose ourselves to an antigen to elicit a primary immune response to generate memory cells so that if we get sick with the actual thing, we skip the primary and go straight to secondary. Now, there are two different types of vaccines. There is subunit vaccines, where only a portion of the pathogen is there. So like for tetanus or diphtheria, you actually just have a antigen spike, so then that's all that they really have. The active version of it is not in there. You can see that it's like vaccines called toxinoids contain inactivated. These ones are just giving you this spike, that antigen spike, or it's giving you the inactivated toxins from it so that you can produce antibodies to the toxin. I also flipped this head to see if they would talk about the other type of vaccine. They didn't, but what it is, it's an attenuated virus, which means that there is a whole functioning virus injected into the vaccine, but it is weakened on purpose. This is so that you can have two different types of responses, but since it's not here, don't worry about it. I'm not going to test you. Potentially a question could be about the subunit vaccines, and quite frankly, I'm going to really be looking for what's the purpose of it. And the purpose of it is to get the primary immune response first so that you can have a much more effective secondary immune response when you actually get sick. Now here, active and passive immunity, we'll talk about it right here. So active immunity means that your body makes antibodies. Now, this could either be through natural exposure, somebody sneezes and you breathe it in, or it's artificially because you got vaccinated. The other one is passive. This is when you are acquired from a mother's breast milk. There are specific antibodies in there, and that helps protect the fetus off the rip, or artificially acquired through injections of antibodies. So in terms of like an antivenom or an antitoxin, you are essentially given antibodies directly into it. You don't make them yourself. And the important thing to remember between active and passive is that there is no memory cells, no lasting protection. Your body, to have that secondary immune response, has to make its own antibodies because it has to have that plasma cell and that memory cell for that memory response.